Good afternoon, everyone. Today we will discuss about the microbial conversion processes for biomass. In this session 6, we will discuss lot about microbial conversion products and the available processes to convert the biomass into useful products by the presence or by the means of microbes. Coming to the introduction for this biochemical process, uh, this microbial process conversion process, it comes under biochemical process because here we are using the biological means like microbes and enzymes, and in the process also there are some chemical conversion, so the whole process indicates biochemical conversion. So, the biochemical conversion is the decomposition method of the biomass using bacteria and enzymes or any other means of living organisms to produce the carbohydrate. This carbohydrate will be further processed to produce sugar from the carbohydrate. The extracted sugar is then used for production of biofuels and other bioproducts through the use of biocatalysts and microorganisms. In the addition of, we are also using heat and other chemicals. These bioconversion examples include alcoholic fermentation, anaerobic digestion, and microalgae based others microbial fuel cell, where we are converting the bioenergy to bioelectricity. The bioproducts from the biomass conversion can include renewable gasoline. Others are various alcohols, acids, so it includes various chemical products, ethanol, and other forms of alcohol, acetates, these things. Despite the importance of the biochemical conversion, which it offers in the production of various biofuels, there are still a lot of difficulties which are preventing it from being commercialized at industrial level. The main uh, limitations for the biochemical process is the cost and the difficulty in breaking down the complex structure of the cell walls of various plant kingdom. And it also has limitation of low hydrogen production after the conversion comparative to the thermal processes. Also, there is high sensitivity of the hydrogenase towards the oxygen. There are technical issues in the genetic engineering, also it adds to the factor, while the biochemical conversion products are not yet being commercialized. So coming to the biochemical process, it also needs biological pretreatment which may help to reach an ideal conversion rate. The technology which is behind the conversion of the biomass is capable to produce various products such as we are getting hydrogen, biogas, different alcohols, butanol and ethanol and other alcohols, organic acids and many others by choosing different organisms during the process of this biochemical conversion. So the microorganism and the type of the microorganism play an important role for our ultimate product. So choosing different microorganism is very important for that aspect. When we talk about biochemical conversion processes, there are some key conversion processes which are we are going to discuss in today's session. First one is the anaerobic digestion process. In anaerobic digestion process, microorganisms they break down the organic matter in the absence of oxygen because the process is anaerobic, and we product of biogas and digested. This biogas is the combination of methane and carbon dioxide, where the digested or the other product byproduct of this conversion process is itself a solid state nutrient rich byproduct, and this can be used as a good fertilizer for the soil fertility increment. So it is also this anaerobic digestion process is used in wider ways for treating organic waste, sewage sludge, and various agricultural residues. The biogas which is obtained out of the anaerobic digestion process can be upgraded to biomethane for use as a renewable natural gas. Coming to the other process of the biochemical conversion, it is fermentation. In the fermentation process, microbes, that's yeast and bacteria, they convert the sugar into various alcohols and other bio-based chemicals, such as ethanol production from sugarcane or corn or lignocellulosic biomass. We can opt the process of fermentation to produce ethanol out of these biomass sources. The key pathway in the bioethanol production and the biobutanol production, we highly recommend the fermentation process. The other process for the biochemical conversion is composting. Composting is controlled way of aerobic biological decomposition of various organic matter into stabilized humus-like compost. We generally do composting for the huge amount of the municipal solid waste. It is used for soil conditioning and waste reduction processes. Enzymatic hydrolysis is other process as biochemical conversion, where the enzymes they break down the cellulose and hemicellulose parts into some fermentable sugars, and this sugar is further processed to produce the biofuels. A crucial step, this enzymatic hydrolysis, it is a crucial step for producing second generation biofuels. The last, the last biochemical conversion process, which will be covered in this session, that is microbial fuel cell. Here, the microorganisms will help to generate the electricity while breaking down the organic waste material. It is obviously a promising bioelectrochemical system for energy generation from wastewater. It also utilized for wastewater treatment in a wider way, but this microbial fuel cell is not yet commercialized because it is a cost effective method as of now. So coming to our first process, that is biochemical process, which is anaerobic digestion method. In the anaerobic digestion method, organic materials are converted into various gaseous mixture directly by some selective microorganism in the absence of oxygen. The produced gaseous mixture they mainly comprised of methane, carbon dioxide, and hydrogen sulfide, which is in a small quantity. It is collectively known as biogas. This anaerobic digestion process is very suitable for organic waste material, which is having higher moisture content of about 70 to 90 percentage. Different biomass sources such as agricultural and algal residues are employed for the anaerobic digestion method. The anaerobic digestion process is advantageous in terms of reducing the biomass odor, its emission of harmful gases, reducing the oxygen demand of the water body, and the production of valuable byproducts such as fertilizer and manure. But we already discussed that this installation cost and the time required for the operation and the maintenance, these are main limitation for this anaerobic digestion. Any kind of biological process, it takes longer time than the thermal processes that we discussed earlier in the last classes where we discussed thermochemical processes. The steps of the anaerobic conversion process are in it is having hydrolysis, acidogenesis, acetogenesis, and methanogenesis. Numerous anaerobic bacteria and fungi, they have been shown to play a crucial role in the anaerobic digestion process. Anaerobic fungi belonging to Neocalimatigo phylum has been promising candidate in enhancing the lignocellulosic based biofuel production. The research have been shown that this kind of this kind of fungi or microorganism, they play important role for production of biofuel utilizing the lignocellulosic biomass. Some other common bacteria include these kind of examples I have presented, for which can refer that these fungi are very useful for conversion of biomass into biofuel. Hydrogenotrophs and the members of methanosarcinaceae are the common bacteria which is being utilized for the acetogenesis process, which is a step of anaerobic digestion. Now you can see some schematic of anaerobic digestion chamber. We discussed that the steps involved in the anaerobic digestion are hydrolysis is the first step, acidogenesis, acetogenesis, and methanogenesis. Here we can see the substrate are being fed into the chamber. It comes down, it mixes well in the presence of a stir tank. So it is mixing well, and in the whole chamber, this many of series of reaction occurs, and the product is biogas as a main product, and it will the gas will go up and it will be collected at the top of the reactor. Here we can see in the digestion process that uh, in the step of hydrolysis, it is kind of pre-treatment of the biomass, right? The conversion of the organic substrate, it is converting into some simple monomers in this step by hydrolysis. These complex biopolymers of various proteins, polysaccharides, fats, or oil, they are breaking down to simple monomers and oligomers in the hydrolysis process. In the second process of acidogenesis, where the acidogenic bacteria, It aids the production of hydrogen, CO2, and volatile fatty acids. This in the second method, in this in the second third method, that is the acetogenesis, where the mainly the volatile fatty acids are again uh, oxidated to acetate, and it takes place in the acetogenesis process. The volatile fatty acid is converted to acetic acid, carbon dioxide, and water vapor. In the last step, where the oxidized acetate substrates are finally converted into methane and carbon dioxide. So at the final, we are getting biogas. Now we will discuss one by one these steps that we have discussed in the schematic. The anaerobic digestion process typically occurs in a closed vessel condition, and the produced biogas it flows out to temporary storage and later on to the end use applications. The main commercial application of the biogas production are heat and electricity generation out of those biogas. After anaerobic digestion process, the vessel will contain the solid residue and organic matter which is known as digestate. This digestate can be separated into liquid and solid streams, and both streams contain valuable plant nutrients and can substitute as fertilizer in the agricultural applications. So the steps of the anaerobic digestions, first step we are going to discuss hydrolysis. So this is very essential pretreatment process of the biomass where the water and extracellular enzymes they break down the complex polymeric structure of the cellulose starch protein and convert them into respective simple units like monomers or oligomers such as glucose, fatty acid, and amino acids. This complex structure of cellulose is converted into glucose is it is converted into monomers. Hydrolytic enzymes generally include amylase, cellulase, lipase, protease, and pectinase. These enzymes are used for in the hydrolysis step. So typically the growth rate of the hydrolytic bacteria is very fast. However, for the lignin-rich substrate, the breakdown of the polymer turns into the rate limiting stage because this lignin is not easy to break. Some compounds in this stage are ready to be converted into biogas, but most compounds need further breakdown through other stages. The next step, that is acidogenesis. At the products of the hydrolysis or the first step is further broken down into the acidogenesis state by the means of acidogenic bacteria. Hydrolytic products are mainly transformed into short-chain volatile fatty acids, which include acetic acid, propionic acid, formic acid, lactic acid, and other alcohol and ketones. We are getting gaseous products CO2, hydrogen, ammonia, alcohols, and trace amount of the other products which are generated as a byproduct. So some of the products such as carbon dioxide and hydrogen acetate and formates, they are readily usable by the methanogens which is being utilized in the last stage. Other products needs to further decompose in the methane decomposed in the next step of acetogenesis. so that it can be utilized in the last stage of methane production. Acidogenesis is generally very fast process, and the risks of formation of this short chain volatile fatty acid accumulation in the digester, it results in digester toxicity if it is not properly controlled. So this product of the acidogenesis, this as this is a very fast process, so we need to control this production of the VFA so that it doesn't create toxicity inside the digester, because this formation of toxicity or this toxicity level, if it goes high, then it will be affecting the other microbes in the other steps. So we can see this glucose of the hydrolysis step is further converted into ethanol, propionic acid and acetic acids. The third step is the acetogenesis. So these acetogenic bacteria, they transforms the product of the acidogenesis stage and some of the long chain VFA from the hydrolysis stage into acetate, carbon dioxide and hydrogen. The reactions associated in the acetogenesis stage are not thermodynamically spontaneous. When, when the partial pressure of the hydrogen is greater than 10 to the power minus 4 atmosphere. Nevertheless, the methanogenic bacteria, it can lower this partial pressure by consuming the produced hydrogen. This syntropic relation, where some bacteria are fed from the other bacteria's product, makes the acetogenesis stage thermodynamically feasible. This interspecies hydrogen transfer is synonymous with electron transfer, as hydrogen is especially a proton donor with an additional electron. The rate of this electron transfer can significantly influence the overall digestion rate. So these are here where we can see that long chain fatty acids are converted into acetates. The last stage is methanogenesis, the final stage where the methane is produced from all intermediate products of the previous stages. This stage is strictly anaerobic in nature, as the methanogenic bacteria cannot survive in the presence of oxygen. Acetate and hydrogen are converted into carbon dioxide and methane by two different groups of bacteria, which is known as acetophilic and hydrogenophilic bacteria. And these acetophilic bacteria convert the acetate into methane and CO2, while the hydrogenophilic bacteria convert the hydrogen and CO2 into methane. So ultimately we are getting methane and CO2 combination, some of the byproduct. So we are now going to discuss some key process parameters of this anaerobic digestion process. As we have seen that intermediate steps, some of the intermediate steps are very fast processes. So we need to precisely control the process parameters to get the maximum output. The sum of the critical parameters are temperature, retention time, organic loading rate, pH and carbon to nitrogen ratio. So the critical process parameters are temperature, first one. So temperature, where two temperature ranges are widely accepted as most favorable temperature range for maximize the performance of the anaerobic bacteria. A digestion temperature range of 55 to 60 degree is defined as thermophilic digestion. The thermophilic digestion, they require additional source of energy or heat input, but it provides a higher biogas production rate. However, thermophilic process is often unstable. A thermophilic temperature increases the feed degradation rate, leading to a shorter hydraulic retention time. When the degradation rate is higher, then the retention time would be lower. So it also generates high quality digestate with fewer pathogens in it. Another temperature range is 35 to 40 degree temperature, which is known as mesophilic digestion. Most of the commercial digesters operate at the mesophilic range. Mesophilic digestion produces less biogas than the thermophilic digestion. However, the mesophilic digestion provides a more stable operation with lower operating costs. So based upon the availability of the biomass, we need to select which kind of temperature zone you are going to operate the anaerobic digestion. Organic loading rate, which is defined as input rate of the organic material per volume of the digester, it is an important parameter for the anaerobic digestion process. Higher than the optimal OLR or the organic loading rate, it can cause over toxicity to the digester, leading to decrease in the methanogenic activity. This toxicity, it may cause the higher accumulation of the volatile fatty acids and many other toxic elements. On the other side, if there is the organic loading rate is lower than the optimal one, then it will reduce the biogas production. So you can see you cannot go higher or you cannot even go lower, because there is some optimal zone where we can get maximized production of the biogas. The next process parameter is hydraulic retention time. The hydraulic retention time, it defines the average duration that the substrate resides in the digester, just like other retention time we generally refer in the any kind of the reactor. These substrates should be allowed to stay in the digester for sufficient time so that to ensure the maximum conversion of the organic material into biogas. However, a longer hydraulic retention time requires larger digester size, which again it will go higher capital cost. So it will be it will be cost intensive. The next one is the carbon to nitrogen ratio. The substrate carbon to nitrogen ratio is another crucial factor which will balance the anaerobic digestion process. Carbon, which is energy source for the microorganisms, whereas they require certain amount of the nitrogen for their growth or metabolism. So, in the digester, the optimal carbon to nitrogen ratio should be around 20 to 30 for most of the anaerobic digestion process. Depending also, it depending upon different characteristics of the substrate that we are introducing. When considering a low carbon to nitrogen ratio, it represents a higher nitrogen concentration, which will eventually turn into excess ammonia production. Excess ammonia produces higher digester alkalinity, and that will inhibit the digester process resulting in lower yield of the biogas. On the other hand, when there is high carbon to nitrogen ratio, which means there is a deficiency of the nitrogen in the substrate, so microorganism will readily consume the nitrogen and not enough nitrogen will be available for their growth or metabolism. So adding nitrogen or carbon rich organic material to substrate will be, if there is no sufficient growth or metabolism uh, of the microorganism, obviously the biogas yield will reduce. So always we need to keep monitoring that the what is the carbon nitrogen ratio of the substrate, so that we are getting a optimal value which is being used in the maximize production of the biogas. The next one is the pH. It defines the concentration of the hydrogen in any solution. That's common definition. Most microorganisms prefer a neutral pH range. Methanogens are very sensitive to pH and it always prefer around neutral or 7 for their highest performance. Hydrolytic and acidogenic bacteria perform between acidic zone like 5.5 to 6.5. On the other hand, acidogenic bacteria more tolerant to larger pH range. But methanogen, they are very sensitive, so they only gives maximum uh, efficient performance in the neutral range of the pH. Maintaining an optimal pH for all kind of microorganism in the same digester is very challenging, especially when the substrate having various compositional variation like sewage sludge. So maintaining an optimal pH is very critical for this case. Now we are going to discuss what are the key challenges that we have discussed lot of process parameters and also the steps of the anaerobic digestions. Now we are going to discuss that what are the challenges we are facing to operate the anaerobic digestion process. First, when the biomass feed biomass stock is having high lignin content, so it will reduce the biodegradability of the substrate due to the recalcitrant nature, which will result in the slower hydrolysis stage. That is the first stage. The second point is attaining the optimal carbon to nitrogen ratio for an effective anaerobic digestion is challenging with many available feedstocks type. Where the four different types of bacteria breakdown we have discussed for the organic material in four stages. Each type of the bacteria they require specific process condition that we discussed last slide, that there is a specific temperature zone, specific pH range and the carbon nitrogen ratio to function properly. Due to the syntropic relation among the bacteria at different stages, disruption in one stage will cascading the effect to the other stage. Means disruption in one stage will affect the production of components or the chemicals in the other stage, so overall production will be low. Higher the accumulation of several intermediate products like ammonia, volatile fatty acids or these kind of intermediate products during the digestion, it is very detrimental effect it shows for the microbes, resulting in the process inhibition. So this kind of intermediate product, they itself act as a inhibitory material. Biogas, which is the main product of the anaerobic digestion process, it has lower calorific value than its natural gas counterpart. The presence of more carbon dioxide, which is 40% around, it reduces the energy content. flame speed and increases the transportation cost. So we are not getting that much of energy content in those biogas. Still we are paying for it that much of volume of the gas for the transportation. So meanwhile we are what we are doing, we are not getting enough energy content, but we are paying extra money for the transportation. So transportation cost increases. Another anaerobic digestion co-product is the unconverted substrate, which is digestate, known as digestate. It is mostly used for the soil application as a replacement for chemical fertilizers due to its nutrients value. High moisture content of the digestate often requires again solid to liquid separation methods for their easy transportation and storage. These separation processes reduce the digested nutrients value. So, meanwhile, a high volume of the digestate is also challenging. So, we can see that how much it is essential for uh, maintaining a optimal range of each of the parameters, so that we can optimize the whole process and minimize these kind of challenges associated with the anaerobic digestion. The next we are going to discuss the fermentation process. So the fermentation is also an enzyme catalyst anaerobic breakdown of the energy rich compounds such as carbohydrate to it break down to carbon dioxide and alcohol and other organic acids by the actions of the microorganisms, especially different bacteria and yeast that occur naturally. The process occurs naturally and it is commonly used in the production of various food, alcoholic beverages, and pharmaceutical products, especially in a controlled microbial enzymatic activity. This anaerobic process occurs without oxygen and starts with glycolysis, which breaks down the glucose into pyruvate. Depending on the microorganisms and their conditions, the pyruvate is further metabolized through different pathways, such as it can follow the ethanol fermentation for producing alcohol and carbon dioxide, or it may follow lactic acid fermentation for producing lactic acid. So the process of fermentation, it involves glycolysis, first step where glucose is broken down into pyruvate, producing a small amount of the ATP and NADH. Anaerobic conditions where fermentation occurs in the absence of oxygen, differing from aerobic respiration. There are different pathways that we have discussed. One is alcoholic fermentation, another one is lactic fermentation. Byproducts are, it is also dependent on the pathway that we are choosing. Fermentation produces specific byproducts like ethanol, lactic acid, acetic acid. The fermentation yields less ATP compared to the aerobic respiration, but it is more important for energy production under the anaerobic conditions. Where we have seen the product utilizations are mainly with different food, alcoholic beverages, and pharmaceutical products and different industrial applications. So now we are going to discuss this pathway that we have discussed. First one is we are getting glucose as a source. It is from glucose. It is being uh, glycolysis by the process of glycolysis. It is being converted into glyceraldehyde 3 phosphate, and from there it is converting into 3 phosphoglyceric acid, where the NAD is reduced to NADH. It is then again it is converted to pyruvic acid. This pyruvic acid follow uh, two method that depending upon what we are choosing. It can produce the lactic acid, this pathway, and another one is the alcoholic, that is the ethanol plus CO2. Here what we are doing, here it is being oxidized and the similar is happening here. So these are the two pathways that it follows from the pyruvic acid conditions. Coming to the next part, that is the fermentation types. So fermentations are of various types depending on the specific microorganisms that we are choosing involving in the reaction and the resulting byproducts. Commonly involved microorganisms in the fermentations are yeast for if you are going for the alcohol fermentation, lactic acid, uh, bacteria for lactic acid fermentation, and acetobacter for acetic acid fermentation. Lactic acid fermentation. So it is a metabolic process by which the starch or sugar is converted into metabolite lactate, which is lactic acid by yeast strains and bacteria. Uh, it is an anaerobic fermentation process that occurs in some bacteria and animal cells where the energy spent is more than the supplied oxygen. In this process, one glucose molecule is converted into pyruvate and then into from pyruvate to lactic acid. This lactic acid production is happening. Alcohol fermentation, ethanol fermentation is or categorized as alcohol fermentation. It is the biological process where the pyruvate, the end product of the glycolysis, this glycolysis is pyruvate. It is broken down into ethanol and carbon dioxide. Yeast performs well in this process in the absence of oxygen. Alcoholic fermentation, obviously, it is an anaerobic process and it is mainly used for the preparation of the alcoholic beverages like beer and other ethanol as a fuel production. We are producing ethanol. The next one is acetic acid fermentation. also known as vinegar. Acetic acid is also known as vinegar. The production of the acetic acid generally involves two processes. First is the involving of yeast for the production of alcohol, which is being done from sugar, and the second one is utilizing the acetic acid bacteria to oxidize those ethyl alcohol to acetic acid through the acetaldehyde method. Fermentation of the acetic acid is done by two methods. One is surface fermentation and another one is submerged fermentation. So these two types of fermentations is going to be discussed in subsequent. So we are getting acetic acid. Butyric acid, it is metabolic process which microorganisms they break down the carbohydrate and produce the butyric as a byproduct. It is this acid is a type of short chain fatty acid, and it finds its application in digestion, gut health applications, and other industrial applications. So we are getting carbon dioxide, hydrogen, as well as butyric acid. So when we are classifying the fermentation processes based on the substrate we are using, first one is the solid state fermentation. Next one is the submerged fermentation. These two broad categories will be discussed. So first one is solid state fermentation. It is defined as the fermentation process which is occurring absence or near absence of the free water utilizing in the process. It is a biomolecule manufacturing process used in the industry. These biomolecules are mostly metabolites generated by the microorganism grown on some solid support, which is this solid support is being selected purposefully. Substrates used for this process can be rice, wheat, straw, wheat bran, or other fruit or vegetable waste. Some organic waste you can take. So, what are the benefits of the solid state fermentation? That is, we are using in the absence of water, so we are having less effluent release and reduced water pollution. Simple and cost effective method. Aeration process is easy for this because it is we can go for the easy aeration in the on the solid supports. It resembles the natural habitat of some fungi and bacteria. So this solid state fermentation uses solid substrate, thus nutrient rich waste material can be easily recycled as a substrate. And the substrate are used very slowly and steadily, so the sum of the same substrate can be used for longer fermentation period. And it is best suited for the fermentation techniques involving fungi and microorganisms that require less moisture content. The next one is submerged category. Submerged fermentation is the method for cultivating the microorganism in a liquid nutrient media. So here we need a media, liquid media, not like the solid support that we discussed earlier. For industrial production, this implies the growing selected microorganisms in some closed vessel called bioreactor which contain the nutrient broths. The system design for the bioreactor allows supply of oxygen as required for the aerobic microorganisms and the ability to monitor and control the different parameters for the process like pH, temperature, viscosity, dissolved oxygen in the nutrient broth, foam formation, biomass formation, substrate utilization, and desired product formation. In the submerged fermentation, the microorganism grown on soluble or insoluble soluble substrate like xylene, pectin, mannan, or insoluble substrate like wheat, straw, rice bran, this kind of, they are dissolved on dissolved or submerged in the liquid media. So these submerged fermentations are more commonly used in the industrial fermentation processes than the solid state fermentation for enzyme production activity due to its ability to control the fermentation parameters using defined substrates. So, however, this submerged fermentation is not practical when we are introducing complex structure of the substrate, such as some agricultural residue or forestry or food processing residues and waste. This substrate, this complex nature needs to be first processed or pre-treatment should be done before it is introduced for the microbial conversion. So there are four types of the submerged fermentation processes available: batch culture, continuous culture and fed culture. Three types. So batch mode represents the fermentation of the sterilized nutrient broth or solution in a closed vessel by desirable microbial culture within that. The advantage of such process is it is low cost, simple infrastructure for the process control. Also, it is having challenges like it causes low productivity and feedback inhibition are the major disadvantages. Whereas the other mode is continuous mode, it involves tightly regulated and high productive bioreactor where the sterile substrate and other medium components are fed at a particular rate and an equivalent amount of the product is harvested in a time dependent manner. So it is more controlled way. Fed batch fermentation is characterized as an intermittent feeding and maintenance of the optimum concentration of required growth substrate with continuous harvesting product. It is having continuous collecting the product out of the reactor. Fermentation types depending upon that we have differentiate between the solid state fermentation and submerged fermentation, where free water is not required for the solid state but it is the main component as a media in the submerged fermentation. Inoculation size, the solid state fermentation require large size more than 10%, but submerged fermentation inoculation size is small. Production rate is high for solid state, whereas production rate is low for the submerged fermentation. Product yield also high for solid state. Mixing is very aeration or mixing is very hard for the solid state fermentation, but it is easy for the media based submerged fermentation. Temperature control is also hard for the solid state because removal of the metabolic heat is hard. But in the liquid medium, it is very easy for the submerged fermentation. State at the end of the fermentation, the medium is a wet substrate with high product concentration, whereas the medium is liquid with low product concentration. Simple extraction processes are followed and less wastewater generated in the solid state fermentation, but complex extraction processes are to be followed, including there is large uh, production of the wastewater. And but we can see that it is very efficient to produce the desired product through the solid state fermentation, the high yield we are getting. But still in the industrial process we are having submerged fermentation because we are operating in a large scale, and there the aerations and mixing is very easy. So here we are following the submerged fermentation at the industrial level. So coming to the bioreactors which are being used in those fermentation methods. So solid state fermentation bioreactors is having generally four groups. Uh, Here one type is the static reactor. Static reactor, it is having the tray bioreactor as an example. We can see this is the schematic for that. It is group 1 reactor where the some trays are there and aeration is passing over the solid state um, chamber. So here it is the static bed condition and mixing is done occasionally with no forced aeration. Second one is the packed bed bioreactor where we can see that bed is either static or mixed infrequently with forced blow air through the bed. So there is occasional agitation without forced, but sometimes there is forced blow air through the bed in the packed bed bioreactor. Air can be passed through from down to top, and the packed is microbes are being packed inside the reactor. The other one is the stirred bed reactor or rocking drum reactor. So here what is happening that cylindrical drums which are being placed horizontally, the where the bed is mixed continuously and the air has circulated within the bed with no forced aeration. It is a slow continuous agitation is happening. And the fourth group is bed bioreactor, a rocking drum bioreactor, also the gas solid fluidized bed. We can see that here the bed is agitated well with the help of forced agitation. The more mixing is happening in this fourth group of this solid state fermentation bioreactor. The next is when we are discussing the submerged fermentation bioreactor. It is the stirred tank fermenter which is being utilized well industrially um, available. Here the steering is being done through the impeller and through the sparger air bubbles are being uh, passed through the this stir tank fermenter. Where in the bubble columns here it is no steering is being done, only the air bubbles are goes upward, and by these bubbles, in the presence of this bubble there will be vigorous mixing in the broth and there will be more conversion. The rate of this bubble rising velocity also plays important factor for the biogas production. The next one is the airlift fermenter, which is two type. One is internal loop airlift fermenter, another one is the external loop airlift fermenter. Here internal one there is aeration opening there is inside, so riser is there and it comes down, liquid flow is happening in such a way. This is riser, inner one is riser and outer one is the downcomer region. So liquid is happening, liquid is flowing in such way. Whereas in the external loop airlift fermenter, the it is not going inside of the separator, it is just following the circular loop. The next one is the fluidized bed fermenter. The fluidized bed, it follows it is well dispersed condition where the fluidized biomass, you can see it is completely fluidized zone is maintained and some of the product is collecting at the top of the fluidized bed fermenter. Next one is the trickling bed fermenter where the nutrient broth is not being in the on hold mode, it is just spraying over the packing, and the air is being fed at the bottom of the packing. So this counter current interaction is happening. So it will maximize the product yield, and the exhaust gas will be taken out from the outlet of the trickling bread fermenter. The next application, that is of the microbial conversion process, that where we are utilizing the the bioenergy to production for the production of the bioelectricity. It is a electrochemical conversion process. Kindly excuse for 5 minutes. Mm. Is there any question as of now? So now coming to the microbial fuel cell, which is also known as electrochemical system. So here the microbial fuel cell, it converts the chemical energy to electrical energy through the reaction of catalyzed microorganism under anaerobic condition. So a typical microbial fuel cell, it consists of anode and cathode just like other fuel cell. This compartment is separated by one polymer electrolyte membrane. The fuel is constituted by organic matter, which can be like glucose and acetate lactate, and this operating temperature is 20 to 35-degree temperature. Like normal ambient conditions also we can do. This type of fuel cell is similar to the polymer electrolyte membrane cell, fuel cell, which is fueled by hydrogen or low molecular weight alcohols, which is operating at a very high temperature of 80 degree. In both of the cases, like when we are considering the PM, MFC or the normal microbial fuel cell. In both the cases, the basic materials are electrodes which are formed by catalyst supported on the carbon substrate and a proton or anion exchange membrane. However, the performance of the MFCs are significantly lower than that of polyelectrolyte membrane cells. The main difference between the microbial fuel cells and the normal existing polymeric electrolyte membrane cells is the anode catalyst. The anode catalyst in the polymer electrolyte cells are commonly platinum or any other platinum based compounds, whereas the anode catalyst used in the microbial fuel cells are the microorganisms. The main technological challenges regarding the microbial fuel cell membranes are biofouling and the internal resistance. The proton exchange membrane, which is separating the anode and cathode chamber, they are more prone to biofouling, which can affect the power output and durability of the MFC. This membrane bifouling normally occurs on the membrane surface facing towards the anode chamber by attachment of the bacteria and other organic substrate to form a biofilm over the that membrane. So it is creating the biofouling. This biofilm acts as a physical barrier for the proton transfer from the anode to cathode, which increases both the pH gradient between the anode and cathode, also deteriorating the bacterial growth and electrical resistance of the MFC. So to maintain top MFC performance, we have to replace the fouled membrane with a new membrane quite oftenly, which may increase the MFC operational cost. So it is cost intensive in such way because it is highly prone to the biofouling. So we can use various types of the waste like food waste, soil or sediment waste, household waste and other agricultural waste or also wastewater also we can use as a substrate for this case. By electrogenic bacteria to we can convert those substrate by electrogenic bacteria to bioelectricity through such MFC. So these are efficient for any bioelectrochemical system which shows up to efficiency 80% in terms of the conversion of the waste matter into energy. So when we consider the basic setup for the MFC, it requires one anode, one cathode, one ion exchange membrane, and organic matter or substrate as a fuel for the growth of microorganism. So based upon the mode of operation, microbial fuel cells are grouped into two categories. One is mediated, another one is unmediated. So what is mediated MFC? That is where the chemical medium is required to transfer the biogenic electron to the anode in the MFC. However, in an unmediated type, there will be redox proteins, which is cytochromes will be present on the outer membrane of the microbial cell, which will facilitate the direct transfer of the electron to the anode. So there, in the mediated one, there will be chemical medium, whereas in the unmediated there will be no chemical medium. There will be there will be some cytochromes which will help to transfer the electrons to the anode. The MFC's methods of electricity generation may be environment friendly, also cost effective, produce harmless product, but currently it doesn't guarantee an adequate amount of generation of energy to serve as a competitive alternative as to the existing technologies. So when coming to the anode chamber, it has anaerobic conditions, while cathode chamber will be having oxygen in presence. Usually, the material used in is looked for the property which will have good conductivity, high surface area that will allow the biofilm formation and which will not be so much corrosive in nature. So the MFC is a type of the green energy conversion technology that produces electricity and has many other advantageous application, such as it will reduce the emission of the toxic compounds. Also, uh, the MFC is used as a biosensor as well. The advantage of the MFC, it increase the high efficiency of the energy conversion, source supplies in abundance along with the mild conditions like mild pressure temperature conditions we are maintaining, ambient pressure temperature condition, and it triggers with the biodegradable nature of the catalyst, fuel and the product. The energy production from the MFC has an advantage to the environment. So you can see the basic normal schematic of the microbial fuel cell. So this is the anode chamber where it is anaerobic in nature, anaerobic condition is maintained, and here it is aerobic conditions is maintained because we are circulating air in this chamber. So coming to the suppose wastewater is our target substrate for this case. So we are what we are passing the wastewater in the anode chamber, and the microbes are attached on the anode chamber. So it will reduce it will produce the CO2 as a byproduct, and hydrogen will be produced as a reduction as a oxidation state. So this hydrogen will this proton will pass through the membrane where the electron will pass through the outer side of the outer circuit. Now in the cathode what is happening, this hydrogen H plus ion is oxidized in cathode chamber and it is producing the water. So basically there is no harmful emissions from this fuel cell. We are getting significant benefits, like we are treating the water, wastewater treatment is done. Also there is production of the water, and CO2 is also taken as a byproduct of the system. So here we can see as a quick recap of the structural description of the MFC. So there is two chamber, anode and cathode, which is partitioned by a proton exchange member. So H plus ion will go from anode to cathode from through this membrane. So this middle one is the membrane. So in the MFC, the microbes remain in the anode compartment as a biofilm over the anode, which produces electrons and protons after oxidizing the electron donor, usually which is organic substrate present in the wastewater. So it will produce electrons and protons. The anode used by the anode is used by the biofilm microbe as the electron acceptor. So it will accept the electron and it will pass the electron through the outer circuit, whereas the available proton will pass through the membrane. So this anode, which is an electron acceptor, it receives the electrons from the microbe after electrons have first passed through the microbe's electron transport chain by NADH to the terminal electron acceptor, then to the microbe's outer side membrane proteins. These many of examples have been presented, which are mostly used in as investigated as a electrogens in the MFC. This kind of microbes are used in the studies for efficient use of the anode compartment. The electron transfer from the anode to cathode through the external circuit produces electric current and generated protons, which will drift over to the cathode through the proton exchange membrane, it which prevents the movement of the oxygen into the anode compartment. So this is only one way direction. Oxygen cannot come into the anode direction. So this kind of membrane exchange is helpful to avoid inhibition of the electricity, uh, uh, inhibition of the oxygen to pass through to the cathode to anode. So instead, cathode is exposed to the oxygen. So air is circulated into the cathode chamber, so that the extra protons can be converted into water. Formation of water. So we are also generating green water. So at the last of this session, we are going to recap some of the microbial conversion products in an overview. So we have seen that microbial conversion is efficient, but it is not yet being commercialized well for many of the biogas production. But for the enzymatic or pharmaceutical application, this may be useful, because here we can use selective microbes for the selective enzymes or pharma- pharmaceutical product production. But comparing to the thermochemical processes, microbial conversion process is very time consuming. Also, the conversion is less efficient in compared to the thermal conversion process. So the products of the microbial conversion process can be broadly categorized into different biofuels, biochemical, biopolymers, and bio-based other materials like biosurfactant, biofertilizers. So biofuels. So biofuels where the microorganism converts the organic matter into renewable fuels through processes like fermentation and anaerobic digestion. We are getting bioethanol, biobutanol, biogas, biodiesel. This biodiesel is going to be discussed in the next session in a more broader way, because this biodiesel production is being high like hot topic now, using the different biomasses. Also the production of the biohydrogen. So these are some biofuels product from out of the microbial conversion methods. So this biohydrogen is one of the interesting byproduct for not byproduct, interesting product for microbial conversion, because we are using the photobiological method. Different biochemicals like lactic acid, succinic acid, acetic acid, citric and butyric acid. So these are with various, these are application is having industrial applications, pharmaceutical application, bioplastic uh, production, also food processing industry. They are having various application of this biochemical produced by microbial conversion method. Different biopolymers also, which are being produced by this microbial conversion, that can be an alternative for the petroleum based polymers like PHA or alkalonates. These are biodegradable plastics are produced by this bacteria, and these species are being mentioned. So selective selection, this selection of this microorganism for your desired product is very essential. Others are polylactic acids, which are being used for which are being derived from the microbial lactic acid fermentations. Xanthan gum as a thickener, it is widely used. Cellulose and this kind of biopolymers are being synthesized by this microbial conversion process, and which is having various industrial applications. Enzymes and proteins, enzymes like amylase, protease, lipase, cellulase, hemicellulase, and also the pharmaceutical other methods like which are being used as antibiotic, vitamins, antibiotics like penicillin, streptomycin, insulin, and other proteins. These are, and vitamin B12 is also can be synthesized through the microbial conversion process. So, in today's session, we have broadly discussed about the microbial conversion processes. So, if there is any question, you can ask. So, in the next session, we will broadly discuss on the biodiesel production through various processes and selecting on the various biomass sources. So, if there is no question, so I would like to end this session here. Thank you all for joining. We will meet in the next session. Thank you.